everyone, and welcome to Chapter 21. This is going to be the last part of the cardiovascular system. This covers blood vessels and circulation. I did cut out all of the slides on here of the major blood vessels. I figured we covered it in lab. I'd rather stick to mostly physiology, but if you need to use those illustrations to I don't know, study with or look at, feel free, but they are not going to be included in this lecture. To start with, there are two types of vessels that we are going to be discussing, and then of course we need to talk about functions. So the first type of vessel is going to be pulmonary. Pulmonary meaning lungs. So this is going to be the vessels that transport blood from the right ventricle through the lungs and then to the left atrium. The other type of vessels are going to be systemic. Systemic vessels are going to transport blood from the left ventricle through all parts of the body and then back to the right atrium. There are 5 primary functions that I want you to know. The first is that the function of the circulatory system, one of the main ones, is to carry blood. Pretty self-explanatory. This is just going to be moving blood from the heart throughout the whole body and then back to the heart. The second is for the purpose of exchanging nutrients, waste products, and gases. So do keep in mind that by going to and through the pulmonary system, we are picking up oxygen, dropping off CO2, and then doing the opposite when we are going throughout the vessels throughout the body. So it is going to be dropping off oxygen and then picking up CO2. The third is to transport substances such as hormones, components of the immune system, molecules required for coagulation, enzymes, nutrients, gases, and waste products, as well as a few other things. So one of the primary things I want you to get from this third function is hormones are going to be transported throughout the circulatory system. Very, very important system in transportation of pretty much anything aside from, say, like neurotransmitters, which are going to be moving through the nervous system. Otherwise, the circulatory system is how anything is moving throughout your body, getting from point A to point B and then back again. The fourth function is going to help regulate your blood pressure. And the fifth is going to be directing blood flow to tissues. There are a few features of blood vessels I would like to go over. These are going to be terms that should look familiar from lab. The tunica intima is going to be that innermost layer. It is made up of endothelial tissue. It has a basement membrane. It is going to have a connective tissue layer called the lamina propria. And then it is going to have an internal elastic membrane, which is going to have fenestrated layers of elastic fibers. Now, if that term fenestrated looks a little bit familiar, it should. We talked about it with capillaries. That term fenestrated is just going to be in reference to having pores or little windows. The tunica media is going to be made up of smooth muscle tissue and arranged circularly around the blood vessel. This is that muscle layer that is in the middle. Usually it's a little bit thicker when it comes to arteries versus veins. This is also where we are going to have vasoconstriction and vasodilation happening. So vasoconstriction, kind of a little self-explanatory. This is where the muscle is going to contract, thus causing a decrease in blood flow. And vasodilation, which is going to be where the muscle is relaxing, and that is then going to open the vessel up to increase blood flow. The last layer is the tunica externa or adventitia. This is a connective tissue on the outermost layer of the vessels. It is going to vary from dense, regular tissue to near the vessel to loose connective tissue that merges within the surrounding connective tissue. Here we have an illustration showing the histology of a blood vessel. We have that outermost layer, that tunica adventitia, that middle layer, which is going to be the tunica media. Again, make sure you know that that's going to include muscle, and the tunica intima, which is going to include everything that is on the innermost layers of the blood vessels. Here is a nice slide showing a cross-section of an artery and a vein, shown with the A versus AV. You can see that the artery is going to be a little bit on the rounder side. This is because there is a higher amount of pressure versus the vein, which is going to have a lower amount of pressure inside of that vessel. Veins have a tendency to look a little bit more on the squashed side, while arteries are going to look a little bit rounder. The other big difference is going to be how big the tunica media is in either. The arteries are typically going to always have this very thick tunica media section, as opposed to veins, which are going to be on a little bit of the thinner side for that layer. All right, so let's move into types of arteries. Keep in mind that arteries serve the purpose of carrying blood away from the heart, and I always like to remember it as away, arteries away. So blood will always be moving away from the heart. Arteries typically will be carrying kind of higher oxygenated blood, but this is not always the case, and I think you know which vessels I mean. So the pulmonary trunk as well as the pulmonary arteries, those are going to carry low oxygenated blood, but in general, arteries are going to have higher oxygenated blood. And then we have these three classifications: elastic arteries, muscular arteries, and arterioles. Now, we will get to these in just a second, but do remember that arterioles are going to be the smallest type of arteries, and they will lead into capillaries. Moving on to the first type of artery, it is the elastic arteries, sometimes called conducting arteries. These arteries are going to stand out due to having larger diameter, so they are going to be wider, if you want to think of it that way. They will have higher blood pressure inside of the vessels, and they will fluctuate between systolic and diastolic. And remember, those terms are just referring to contraction versus relaxation. They are going to be more elastic tissue than muscle tissue. And then down here, this, I don't know why it's wrong on here, but it is going to have a relatively thick tunica intima, so that innermost layer, and it will have a thinner tunica adventitia. So sorry for that little mistake there, but it is a thinner tunica adventitia than other types of arteries. The second type of artery is the muscular artery. These are going to also sometimes be called the medium arteries. So again, they're going to be a little bit smaller in diameter than the elastic arteries, but they will be larger than the arterioles. They are going to have smooth muscle that allows the vessel to regulate blood supply by constricting or dilating. Most of the smaller unnamed arteries are going to be classified as muscular arteries. The thick walls are due to 25 to 40 layers of smooth muscle. They are also called distributing arteries because smooth muscle allows vessels to partially regulate blood supply to different regions of the body. When it comes to the smaller versions of muscular arteries, they are going to be adapted for vasodilation and vasoconstriction. You can kind of make a little note here that due to that is where we are going to get blood pressure, and it's also going to be what moves blood through our capillaries. All right, into arterioles. Again, these are the smallest arteries, and this is going to cause the three tunics, so those three layers that we've been discussing, they aren't going to be able to be differentiated because they are just so small. They are going to transport blood from small arteries into the capillaries. So again, these are going to be the precapillary arteries. And like small arteries, they are capable of vasoconstriction and dilation. And again, because of that, when it comes to the medium and smaller and arterial arteries, this is going to be pushing blood through the capillaries. And on to capillaries. Capillaries are going to have a wall that consists of an endothelium, so simple squamous epithelium, a basement membrane, and a delicate layer of loose connective tissue. It is going to be scattered with precapillary cells that consist of fibroblasts, macrophages, or undifferentiated smooth muscle cells. Substances are going to move through capillaries by diffusion, and what this means by through the capillaries, literally through the walls of the capillaries themselves. So if you need a little recap of what diffusion is, that is moving items, however you want to think of it, aside from water, because that is a different process, that is osmosis. But diffusion is going to be the movement of particles or items from areas of high concentration to areas of low concentration. So make sure you know that it is high, if I could get my arrow to stay, to low. So make sure you know that. They are going to be lipid soluble, so lipids are fats, just as a little reminder, and small water soluble molecules will be able to pass through the plasma membrane of the walls of the capillaries. The larger water soluble molecules are going to pass through fenestrae, or windows, or can also be called gaps, between endothelial cells, but I like to think of them as windows, if you will, or they can just kind of be like holes. There are 5 types of capillaries that I want you to know: continuous, fenestrated, sinusoidal, sinusoids, and venous sinuses. So the first one is continuous. This means that there will be no gaps between the endothelial cells, no little windows, no fenestrae, and they are less permeable to large molecules than other capillary types. For example, you'll find these types of capillaries in muscle tissue and nervous tissue. So make sure you know the examples of where these types of capillaries will be located. Fenestrated are going to be endothelial cells that have numerous fenestrae, which windows or gaps. The fenestrae are areas where cytoplasm is absent and plasma membrane is made of a thin, porous diaphragm. It is highly permeable, so I would make sure to know that it is highly permeable. And examples of where you'll find this type of capillary is the intestinal villi, the ciliary process of the eye, the choroid plexus, glomeruli of the kidneys, and we'll figure out what that is here in a bit, but just know that it's an example of the fenestrated types of capillaries. The next is the sinusoidal. This is going to be a capillary that is larger in diameter with large fenestrae, so large gaps. It will have less of a basement membrane. An example of where we'll find this is endocrine glands. The next is sinusoids. These are going to also be large in diameter, and they are going to have a sparse basement membrane. Examples are going to be the liver and bone marrow. And then the last type, the venous sinuses, are similar in structure to sinusoids, but they are even larger than those. An example of where you'll find this type of capillary is the spleen. This is a lovely little illustration showing 3 types of capillary walls. The continuous capillary wall is, as the name implies, continuous. It is going to not have any fenestrae, no gaps, while the fenestrated capillaries are going to have these little gaps between within the wall. We do see the different types here with the fenestrae with the diaphragm and without. So there is going to be a little bit of something between those gaps, but not much. We can still have passage of items going through there, while the fenestrae without a diaphragm is not going to have anything to halt anything, if you will, through these gaps. All right, moving on into capillary networks and the vessels that are going to be associated with those. So remember that arterioles are going to be the smallest arteries that we have. The arterioles are going to kind of branch off, if you will, which is going to be that kind of main area going into the capillary bed, and that is the meta-arteriole. The meta-arterioles are going to be kind of met with these sphincters that you're going to see. They kind of look like little rubber bands, if you will. And you'll notice that the majority of these, in fact all of them as far as this illustration goes, is going to be met with these little bands of muscle. Those are the precapillary sphincters. So they are going to be these little smooth muscle bands within the arterioles and meta-arterioles. So that is going to be what regulates that blood flow going into the capillary network. From the precapillary sphincters, we are going to be going into this area of kind of gas exchange happening. So we're going to be dropping off oxygen, picking up CO2, and then we are going to leave those capillaries within the venous capillaries and then out through the venules. Next up is the Arteriovenous Anastomoses. These are just going to be vessels that are outside what we have seen thus far. So they don't necessarily fit into exactly arteries, don't exactly fit into veins, but they are specialized in a lot of ways. The Arteriovenous Anastomosis is going to be a specialized vascular connection that allows blood to flow directly from arterioles to veins, and the main thing that you need to get out of this is that we're just going to be skipping the capillaries. So these are just going to skip over those networks and go straight from artery to vein. So the smaller vessels, of course. We are going to have another type of structure here called a glomus. This is an arteriovenous anastomosis with a little bit of a twist. They are going to have an abundantly smooth muscle within the walls of this vessel. They are going to be larger in diameter than venules, so they are going to be a little bit bigger. They are going to be more abundant. You're going to find more of them in the sole of the foot, the palm of the hand, the terminal phalanges, so further out at the fingertips, and in the nail beds. They are going to serve the purpose of regulating body temperature by adjusting the blood flow through them. So what's kind of interesting about these is if you've ever noticed on an elephant how big their ears are and how they flap them around, what this is doing, and I'm trying to draw you a cute little elephant here, what this is doing is it's going to circulate the blood through the ears, and this is going to cause cooling. So the heat is going to be going out and cooling off the elephant via those large appendages that are at the extremities. Glomuses are kind of similar in that way, is that they are going to help regulate the temperature in that area, as well as the temperature of your blood. All right, so we are entering into the types of veins, getting away from arteries. Do you remember that veins are going to carry blood toward the heart? The three types that we're going to be talking about are venules, small veins, and medium or large veins. Those are clumped together. Do keep in mind that when it comes to veins, the walls of these vessels are going to be thinner than arteries, and that is because they have less of a tunica media, so that little muscle layer surrounding the vessel. So venules are going to drain the capillary network, as we were looking at before with the capillary network that we had that illustration of. All that blood is going to go through the arterioles, the meta-arterioles, and enter into the little venules and whatnot. So they are going to be draining that area. Venules are going to be made of endothelial cells and a basement membrane with a few smooth muscle cells. And as the diameter of venules increase, the amount of smooth muscle will also increase. So it is going to be simpatico, essentially. Like, you're going to see one thing increase, the other one is also going to follow. Moving on to small veins, these are going to be composed of smooth muscle cells that form a continuous layer. The addition of the tunica adventitia is going to be made of collagenous connective tissue, and these small veins will be larger in diameter than venules, so do keep that in mind. So venules are still that smallest vein vessel. Small veins are just a little bit bigger than that. Nutrient exchange is going to occur through the walls of small veins, especially close to the intestines, and we will get to that in a little bit. Moving on to medium and large veins, this is going to be the go-between between small veins and large veins as far as medium veins go. Large veins are going to be larger than medium veins. The tunica intima is going to be thin. It is going to be composed of endothelial cells that are going to have a relatively thin layer of connective tissue and a few scattered elastic fibers. The tunica media is going to have a circularly arranged smooth muscle cells layer, and the adventitia is going to be the most predominant layer, making up these larger veins. As we talked about in lab, the veins themselves are going to have a structure that arteries do not, and that is valves. The purpose of the valves is to create blood flow toward the heart and prevent backflow. And you can see from this illustration the way that these valves are going to look when they are open versus when they are closed. They will only be found in veins greater than 2 millimeters in diameter, and the valves themselves are going to be folds of that innermost layer, the intima, to form two flaps that are going to overlap. There are going to be more valves in veins of the lower extremities than in the veins of upper extremities, and this is going to have to do with gravity as well as the way blood kind of moves through those lower extremities, and we'll talk about that in just a little bit. So here we have other types of vessels, much like the Arteriovenous Anastomoses. These are going to be vessels that kind of just don't fit in a normal sense of the other vessels that we have talked about. The first is going to be portal veins. Portal veins are going to be veins that begin in a primary capillary network, extend some distance, and end in a secondary capillary network without a pumping mechanism. For example, the heart, between the two. There are three examples of this: the hepatic, the hypothalamo-physical, and then the renal nephron, so in the kidneys. So the one that we are going to talk about in this lecture is going to be the hepatic, but the other ones, don't worry about those for now. The other type of vessel is going to be the vasa vasorum. This is going to be blood vessels that supply the walls of arteries and veins. They are going to penetrate the vessel walls from the exterior, and they are going to be branches of arteries. And for arteries and veins greater than 1 millimeter, this is where you're going to find them. Nutrients can't diffuse from the lumen to all the layers of the wall, so that is where these vessels come in and kind of do an assist with providing nutrients. All right, so let's talk about how we have neural innervation of blood vessels. Do make a little note that the walls of most blood vessels are going to have unmyelinated sympathetic nerve fibers form a plexi in the tunica adventitia of those vessels. This is going to be what causes vasoconstriction. So we are going to have that tightening of those vessels through this process here. Vasoconstriction is going to be caused by sympathetic stimulation. Small arteries and arterioles are going to be innervated to the greatest extent, and the kind of contrary to that, vessels of the penis and the clitoris are going to be innervated by the parasympathetic. So what this is going to cause is dilation as opposed to constriction, which means that there's going to be more blood flow, which makes sense in those areas as they are vessels and organs that are going to be engorged with blood at various times, specifically during arousal. So some blood vessels are going to be innervated by myelinated fibers and act as baroreceptors, and we will talk about baroreceptors in a little bit, but you can make a little note that these are going to be stretch receptors. They are going to be responding to changes in pressure, and those are going to monitor stretch and detect changes in blood pressure, as I said. So just make a little note of that. As we age, our arteries and veins are going to change a little bit. Here we are going to talk about arteriosclerosis, which you can make a little note that it is hardening. Arteriosclerosis is just a hardening of these vessels, and the more it will increase the work of the heart, essentially. So increase the work of the heart because of a decrease of circulation. It will be harder for blood to circulate, meaning the heart will be working harder to make up for that. The next we have arteriosclerosis, there we go, and that is the deposition of plaque on the walls. And a few things that can cause this is essentially age and various other risk factors. So this is going to be affecting primarily the coronary arteries. What that plaque is going to be macrophages containing cholesterol, and it's going to accumulate in that tunica media. And what essentially is going to happen in the end is a narrowing of the lumen to the point of potentially blocking off or again making the heart work harder, which is not a good thing. That is not what we want. Do make a little note that when it comes to these various conditions, as well as pretty much any conditions that can happen to the vessels, some people are just going to be more susceptible to changes in the body than others. Everyone kind of takes care of themselves in different ways, different diets, different exercises, different requirements for their body, you know, various things. So everyone's going to respond a little bit differently. But for the vast majority, most change that's going to occur over time in the body is going to occur in the large elastic arteries, such as the aorta and arteries that go to the brain, as well as the coronary arteries. Do make a little note that muscular arteries are going to experience change too, but to a much lesser degree than the elastic arteries. All right, moving into the pulmonary circulation. Again, we have talked about this pretty extensively when it came to lab. This is going to be where blood is traveling from the right ventricle into the pulmonary trunk. The pulmonary trunk is then going to divide into the left and right pulmonary arteries, enter into the lungs. That blood is going to become oxygenated and drop off that CO2. And then the two pulmonary veins are going to exit each lung and enter into the left atrium. So again, we have gas exchange in the pulmonary circulation. We are picking up oxygen, dropping off CO2, and then returning to the heart to then have that blood spread throughout the body. When it comes to the systemic circulation, this is going to be split up into the arteries and veins. It can't just be kind of clumped together like we did with that pulmonary circulation. We have talked about all of these lovely things, so I am going to just very briefly say that this slide is going to cover the arteries that we have already talked about, while this slide is going to cover the systemic circulation of primary veins. So remember, arteries are going to carry blood away from the heart, while veins bring blood back toward the heart. Typically, when it comes to the systemic circulation, you can go by arteries are red because they are carrying highly oxygenated blood, while veins are going to have low oxygenated blood and a little bit more CO2. All right, moving on into the hepatic portal system. This is going to be the system that carries blood drained from capillaries within most of the abdominal viscera, so those abdominal organs, to a series of dilated capillaries, which we talked about these before, but they are those sinusoids within the liver. They will deliver nutrients and other substances absorbed to the liver, as well as possibly toxic substances. And we do talk about here where, let me put a little star by it right here, where nutrients are either going to be taken up and stored or modified chemically and used by other parts of the body. In this instance, the liver itself is going to be doing all that work. It is going to be modifying various things to either be used or removed or stored or changed. And so this process is going to be biotransformation. It's where we are going to be taking toxic substances that can be broken down by hepatocytes, so those are going to be liver cells, or can be made water soluble. And this is to be transported in blood and then excreted by the kidneys in the form of urine. Moving on to the dynamics of blood circulation, this is where we're going to have blood movement determined by various interrelationships between these three things. So flow, resistance, and pressure. These three things are the same things, I know I keep saying things, as water through pipes are kind of restricted by. So if you get a little bit confused, just imagine that blood is essentially just water flowing through pipes. It's just a fluid flowing through a tube, if you will. The control mechanisms that regulate blood pressure and blood flow, which we are going to be talking a lot about here in just a moment, are critical to function of the circulatory system. This is also how we are going to maintain homeostasis. I know everything goes back to homeostasis, always, 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 but that is the purpose of the body and the various systems, is to keep you in homeostasis, that optimal internal state. Okay, so let's talk a little bit more about flow, specifically two types of flow that we're going to see within our vessels. The first is going to be laminar flow. So fluids, including blood, tend to flow through long, smooth, walled tubes in a streamlined fashion, which is laminar flow. So keep in mind and underline this word here, which is streamlined. That's a very important thing. If it is not streamlined, you're going to get something called turbulent flow. This is where that flow is interrupted, possibly by fluid passing through a constriction or a sharp turn or encountering a rough surface. So back to laminar flow, it's going to be where the fluid behaves in a tube as if it were made of layers. And so this does talk about those layers a little bit here with the laminar flow. The outermost layer is going to be moving the slowest. So I'm going to try to draw this for you. So pretend that's a circle, not an oval. And so this outer blue layer, if you will, is going to be what is moving the fastest. And imagine that this tube is just full of blood, all right, or some type of fluid. You can think of it as any type of fluid. But this fluid that is in blue is going to be moving, the slowest, this outer layer. While the innermost layer, so all of the fluid that is in the center, is going to be moving faster. It is faster at the center and slower on the outside of that tube of fluid. Now, when it comes to turbulent flow, however, this is going to be where you're going to have this situation interrupted. So again, laminar flow is going to be more streamlined. Turbulent flow is an interrupted flow, and that can also be described as the rate of flow exceeding its critical velocity. And two things I want you to make a little note of down here is that turbulent flow is going to be partially responsible for heart sounds as well as the sounds that you can hear when you are listening underneath a cuff while taking blood pressure, which is kind of cool. So when you think of this turbulent flow, these sounds can be caused from friction in the arteries, and possibly constriction, but essentially it's going to be vibrations from the liquid as well as the vessels surrounding the liquid, and that is what you're listening for when you take someone's blood pressure. All right, and of course we have this lovely illustration that is showing the difference between laminar and turbulent flow. We're going to be encountering some type of constriction there, which is going to cause that turbulent flow, while the laminar flow is showing that nice layered effect of the fluids. And remember, the outside is the slowest, the inside is going to be the fastest of that fluid within that long streamlined tube. Moving on to blood pressure. Blood pressure is going to be that measure of force exerted by blood against the vessel walls. The blood is going to move through vessels because of blood pressure. That's a very important thing to kind of make note of. And blood pressure can be measured by an instrument called a mercury manometer, which is probably the coolest sounding instrument ever. But what this is going to measure is in millimeters. It is going to measure how much mercury can be lifted as a column. So let me phrase that a little bit better. So it's going to measure in millimeters of mercury blood pressure of 100 millimeters of mercury if it is great enough to lift a column of mercury 100 millimeters. So it is just measuring how much that blood pressure could be if it is great enough to lift a column of mercury the same amount, is all it's saying. How it can be measured is directly using a tube called a cannula into the blood vessel, or indirectly, this is how most people do it. This is how you would do it during an examination. This is what happens more often than not, which is the auscultatory, again, you know, I can't say things half the time, method. So this is the normal one with a cuff and sometimes a stethoscope. So this method, which I'm just going to call it this method because, you know, words are hard to say, is going to use a sphygmomanometer and a stethoscope in order to listen for a Korsakoff sound that is going to be produced, again, as I said before, by turbulent flow in arteries as pressure is released from a blood pressure cuff. So just a few things about that. The cuff is going to inflate until the brachial artery collapses or is collapsed. Pressure in the cuff is going to be slowly lowered, and if you can imagine getting your blood pressure taken, I'm sure you can imagine what I'm describing here. So they're going to be releasing that pressure from the cuff very slowly. It is going to decline below the systolic pressure, and blood flow through the constricted area during systole, remember ventricle contraction, is going to happen. The flow will become turbulent, which is going to cause vibrations in the blood and the associated vessels, and that is the sound that we're listening for, that corticoff sound. Now, this just goes through everything a little bit more in detail. In order to keep this set of notes a little bit shorter, you can go through it at your own time if you'd like to. All right, moving into some lovely equations. So this is going to be how we measure flow, as well as a few other things, and then how we're going to combine it to put together a law, which is kind of cool. So bear with me for a second. So the rate of flow through a tube is expressed as the volume that passes a specific point per unit of time. So this can be like, I don't know, a liter per 20 minutes. You know, that's obviously kind of a random putting together of you know, rate if you want to, but you can imagine it. For this example, we're going to be looking at cardiac output at rest being 5 liters per minute, and thus blood through the aorta is going to be 5 liters per minute. So that is what we are measuring in here, is liters per minute. So in order to express this flow mathematically, we do have this little equation here, which is flow equals P1 minus P2 over R, where P1 and P2 are pressures in the vessel at points 1 and 2. R is going to be the resistance to flow, and it is going to always be where you have flow going from areas of high to low. So always think of diffusion. This is going to come up quite a lot, and typically you can always go by that rule that something is going to be moving from an area of high to low. So this is going to be directly proportional to pressure differences, and inversely it is going to be proportional to resistance, which resistance is going to be this lovely equation down here. Just make a little note of it for now. But do make a little note that as resistance increases, our blood flow will decrease, and I'll write that out here. So as resistance increases, blood flow is going to decrease. When it comes to our lovely little equation of resistance, a few things we need to go over. There is viscosity. So V is for viscosity. L is going to be the length of the vessel, and R is going to be the radius of the vessel. And resistance is going to be affected by specifically blood viscosity, vessel length, and vessel diameter. So as long as these things are different, down here, that is going to affect directly resistance. All right, so let's combine the equations that we were looking at before for flow and resistance, and that is going to create Poiseuille's law. So what Poiseuille's law is going to say is that flow decreases when resistance increases, and the reverse is also true. And since resistance is proportional to blood vessel diameter, constriction of a blood vessel increases resistance and thus decreases flow. So as long as you kind of know that, you can use this equation, and I will not ask you to use it, just understand what it's saying. So here it is talking about during exercise, your heart will beat with greater force, which is going to increase the pressure in the aorta. We can imagine that, right? And the capillaries to your skeletal muscles are going to increase in diameter. So they're going to dilate and allow more blood in. This is going to decrease resistance and increase flow. Your increased flow in your aorta can go from 5 liters per minute to 5 times that amount while exercising. So that is just showing a nice little example there of Poiseuille's law. So let's talk about viscosity for a hot minute. So viscosity is going to be the measure of resistance of liquid to flow. So how much resistance you're getting with that liquid. It is going to be determined by considering the viscosity of distilled water as the number 1, so its unit is just 1, and comparing the viscosity of other liquids with that distilled water. So that is how we are getting this measurement of viscosity. Its resistance is going to be proportionate to its flow, and as viscosity increases, pressure is required to flow increases. Viscosity is going to be influenced largely by hematocrit, and your hematocrit is just going to be that percentage of the total blood volume composed of red blood cells. And you can definitely see a little kind of correlation here. So if your hematocrit increases, then your viscosity will also increase. So as hematocrit increases, viscosity will increase as well. Dehydration and or uncontrolled production of red blood cells can also lead to an increased viscosity, which makes sense because hematocrit is going to be the percentage of red blood cells. Makes sense, right? And it can lead to an increased viscosity, which will increase the workload of the heart. That is not going to be a good thing. So too much workload for your heart can equal heart failure, which is not a good thing. But a little example of how hematocrit can affect viscosity is blood with a hematocrit of 45% will have a viscosity 3 times that of water. Another example is 65% hematocrit will create a viscosity 7 to 8 times that. So you can see that it increases logarithmically. Next up, let's talk about critical closing pressure, or Laplace's law. So critical closing pressure, I want you to think of shock, and shock is going to be where your blood pressure ends up dropping rapidly, probably because you're losing blood, right? So your body goes into shock. That blood pressure can't be maintained. There's not enough blood going to your organs. Things are starting to shut down. But that critical closing pressure is going to be the pressure at which a blood vessel collapses and blood flow stops. So blood pressure can drop below your CCP, so that critical closing pressure, and causes vessels to collapse and blood flow to cease, thus causing shock, right? Laplace's law is going to state that the force acting on blood vessel walls is proportional to diameter of the vessel times blood pressure, and we can see that in this little equation here. Thus, as the diameter of a vessel increases, the force of the wall increases, and weakened parts of the vessel wall will bulge out. This is what's known as an aneurysm, and you've probably heard this term before. This is going to be an example of a positive feedback that can proceed until the vessel ruptures. This is often fatal if it happens in the brain or the aorta. So very not good thing. So you've probably heard that term before. Now you know what it is. The next thing we need to talk about is the idea of compliance. And compliance is going to be this relationship between volume and pressure. And it states that as volume increases, pressure will also increase. This is specifically going to be a little bit easier to understand with this little example that I have here. So the more easily the vessel wall stretches, the greater its compliance will be. What I want you to think of is a plastic water cup versus a water balloon. With the water balloon, you're going to have a lot more stretch. So its compliance is going to be greater than the plastic cup, if you will. It's able to stretch and hold more. The venous system, when it comes to compliance, is going to have a larger compliance than the arteries, 24 times greater in fact, and it's going to act as a blood reservoir because of it. Now, of all the blood in your body, about 84% of that blood is in your systemic system vessels. So systemic versus pulmonary, right? So 84% of your blood at any given point in time, roughly, is going to be in that systemic system. 64% of that blood is going to be found in your veins, while 15% is in your arteries and capillaries. And that is because your veins are able to have a larger compliance in your arteries and thus be able to just be little reservoirs. We are going to have this term up here, vascular compliance, and that is a tendency for blood vessel volume to increase as blood pressure increases. So you can kind of think of the fact that veins are going to have a higher vascular compliance than your arteries because they can hold more and stretch like a water balloon. Moving on to the physiology of the systemic circulation. So keep in mind that the primary function of your systemic circulation is going to be distribution. So moving oxygen and carbon dioxide, nutrients, and hormones. What we're going to see here is a term called cross-sectional area. And what this is going to be talking about is as a diameter of a vessel decreases, the total cross-sectional area increases and velocity of blood flow decreases. And this is kind of going to be a weird concept to think of, but we can't look at essentially like every single vessel and figure out the pressure. So what we do is we look at the total cross-sectional area of all of the vessels that match that particular vessel. In this example that it gives you is there's only one aorta with a cross-sectional area of 5 centimeters squared. Comparatively, if we look at capillaries, the total cross-sectional area of millions of capillaries within your body, if we add them all up together, is going to be roughly 2,500 centimeters squared. So yes, your aorta is going to be larger, but there is only one, so its small cross-sectional is going to be smaller than the capillaries, which are smaller in size, but there's about 10 billion of them in your body, and combined, their cross-sectional is going to be larger. So this does give you a little example on this slide, which says, much like a stream that flows rapidly through a narrow gorge, but flows slowly through a broad plain. Do keep in mind that blood flows through vessels at different velocities, and this is going to determine the cross-sectional area for all blood vessels of a given type. As I said, there's only one aorta, but there's millions and billions of capillaries, right? All right, so let's look at pressure and resistance. What I want you to get from this slide is that there is going to be a drop in pressure and resistance, so specifically being called pulse pressure, and you can see in this get a little color here, this little graph that we're going to see this nice little dip happening and that we have our greatest drop in pressure occurring in the arterioles, which are going to regulate blood flow through the tissues. So right here. Blood pressure is going to be changing as blood moves from one vessel type to another, and I love the way that this is showing that movement of blood from our aorta, arteries, arterioles, capillaries, and into venules, veins, and the vena cava. So moving into that pulse pressure, this is going to be the difference between systolic and diastolic pressures. Remember, that's just going to be contraction versus relaxation. But we're going to see an increase when the stroke volume increases or vascular compliance decreases. Compliance is going to tend to decrease with age, which we've already talked about, which is arteriosclerosis, and pressure rises. Pulse pressure can be used to take a pulse to determine the heart rate and rhythmicity, and we can find them in all these areas on this illustration here. But the ones that we're typically going to use more often than not are going to be that common carotid artery that's going to be just underneath the jaw and kind of to the lateral sides of the neck, and then the radial artery, so kind of just below where your thumb is in accordance to your wrist. So as blood is ejected from the left ventricle into the aorta, it is going to produce a pressure wave or a pulse that is going to travel through your arteries, and that rate of transmission is going to be 15 times greater in the aorta as it is going to be anywhere else. But that, just so you know what is causing this pulse pressure, as well as your pulse in general, is just those waves beating through your blood, essentially. Kind of cool. Okay, so capillary exchange is going to be the movement of substances into and out of capillaries. This is going to occur by means of diffusion. Remember, and I cannot repeat this enough, when it comes to diffusion and kind of the movement of anything, it's going to go from an area of high concentration to an area of low concentration. So oxygen is going to move out of the blood and into that interstitial tissues and cells and various things around those capillary networks via diffusion, because there's going to be less oxygen in those areas. So oxygen is going to move out of the blood and into those tissues, while CO2 is going to move into the blood because there's less CO2 in the blood at that moment. The same goes for kind of everything else that is going to be moving into and out of, and you can see a few examples of those there. Blood pressure, capillary permeability, and osmosis are also going to affect the movement of fluid from capillaries. And then fluid is going to move out of capillaries at arterial ends, and most but not all are going to return to capillaries at the venous end of those capillary networks. And what remains in the tissue is going to be picked up by the lymphatic system and returned to the venous circulation. We will talk about the lymphatic system in the next section, but just make a little note here that not all fluid, in fact, I believe the number is about 15% of the fluid and various things that are moving through the capillaries are going to be left in that interstitial tissue. It cannot be left there. We have to pick it up, and we do that with the lymphatic system. Okay, so moving on into a little bit more of what's going to turn into an equation, but we're kind of building up to it. So net filtration pressure, or NFP, is going to be the force responsible for moving fluid across the capillary walls. So this is specifically the mechanism for how we are moving fluids to and fro from the capillaries. there are going to be two forces that will affect the NFP, and that is hydrostatic pressure, which is the physical pressure of blood flowing through the vessels or of fluid in the interstitial spaces, and then osmotic pressure. Anytime you see osmotic, osmosis, anything like that, immediately think water. But that is the movement of solutes through a membrane in the presence of a non-diffusible solute. So large proteins are not going to be able to freely pass through the capillary walls, and the difference in protein concentrations between the blood and interstitial fluid will be responsible for osmosis. Again, movement of water. So moving on into kind of putting all of that together to look at what will be an equation. But we've got our net filtration pressure, our NFP, which is going to be equal to our net hydrostatic pressure minus the net osmotic pressure. So what we were just talking about before. The net hydrostatic pressure, how we get that, is going to be taking our capillary hydrostatic pressure and minusing the interstitial fluid hydrostatic pressure. And that is going to be the pressure exerted by the interstitial fluid of tissue surrounding the capillaries. So think about, I don't know, the capillaries going into these areas and being pushed upon by the tissues surrounding it. That tissue is going to be full of fluids, if you will. So that interstitial fluid, which can be water, it can include hormones, lipids, amino acids, all kinds of stuff, nutrients, oxygen, various waste, etc. It's going to be this IHP is going to be the pressure of that fluid within the interstitial fluid pushing on the capillaries, if you will. Our net osmotic pressure is going to be our blood colloid osmotic pressure minus our interstitial fluid colloid osmotic pressure. Okay, moving into the functional characteristics of veins. So venous return to the heart is going to increase due to increase in blood volume, venous tone, and arterial dilation. Let's briefly talk about what venous tone is, and that is that continual state of partial contraction of the veins as a result of sympathetic stimulation. What I do want you to kind of like look at is the relationship between sympathetic stimulation and venous tone, and it is as sympathetic stimulation is increasing, so does venous tone. This is going to be caused by causing the veins to constrict more, which is also going to be moving the blood, right? So as those vessels are constricting, that venous tone is constricting, it is going to keep that blood moving. So blood pressure and the effect of gravity. I mentioned this just a little bit briefly before when we were talking about vessels in the legs. So in a standing position, the hydrostatic pressure caused by gravity increases blood pressure below the heart and decreases pressure above the heart, which makes sense because everything's going to be moving with gravity. So down, right? It's going to be pushing a lot of that blood towards your feet. The muscular movement is going to improve that venous return, meaning as long as you're moving and not just standing there nice and still, that is going to improve how much blood is going to be returning to your heart. So the right atrium blood pressure, I want you to think of it as 0 millimeters per mercury, while it's 100 millimeters per mercury in the aorta. And while standing, the blood pressure in your feet can be about 90 millimeters of mercury instead of 10 when you're not standing. So this is going to show effects in your legs and feet as far as gravity goes, what it's causing. And that is high pressure from standing, which can be called an edema, and we'll talk about that a little bit more with the lymphatic system. But this is just going to be blood pooling in the lower extremities, and this is why there are more veins in your lower extremities, so that you can give a little bit more in the way of holding blood in those lower extremities. So remember, compliance. There are going to be a few mechanisms that control blood flow. Specifically, we are going to control the blood flow in the tissues. There are 3 types that we are going to discuss a little bit more of: local control, nervous system control, and hormonal control. So local control, what I want you to know is that blood is not equal in all tissues. You are going to have an increase in blood flow with the brain, the liver, and the kidneys. And this makes sense because there needs to be a lot of blood going to those areas, either to be processed, filtered, or to supply important tissues with a lot of oxygen, right? Areas where you're going to have lower blood flow is going to be like the skeletal muscle tissue. You don't need as much going there when you're not using it, but of course, obviously, if you're exercising, you're going to need a little bit more blood going to those areas to take away waste and bring in oxygen. So starting with local control, this is going to be found in most tissues. Blood flow is going to be proportional to metabolic needs of the tissue. This is going to be achieved by periodic relaxation and contraction of precapillary sphincters. So allowing more blood in, not allowing more blood in, just depends on that area's need of blood and oxygen and nutrients. The nervous system is going to be responsible for routing blood flow and maintaining blood pressure, while hormonal control is going to be a sympathetic action potentials that stimulate epinephrine and norepinephrine. And again, we will talk about these more in just a moment. The functional characteristics of a capillary bed, this is going to be affected by local factors that regulate meta-arterioles and precapillary sphincters. Remember, those are going to be on the artery side of the capillary networks. This is going to be causing an increased metabolism of a tissue, resulting in a vasodilator substance. For example, carbon dioxide, lactate, and potassium being too much in abundance, it will cause vasodilation and relaxation of sphincters. Blood flow will increase and to serve the working tissue. A lack of nutrients can also result in increased blood flow to a tissue as well. So an increase in rate metabolism within the tissue will cause an increase in blood flow. So as the need arises, you're going to try to meet that need, if you will. So this is a nice little diagram that is showing that local control of blood flow through the capillary beds. You can read it at your own time if you want to. I'm not going to test you over the exact mechanism outside of the last slide. All right, moving into autoregulation of blood flow. What you can kind of write down about what is autoregulation, this is just the maintenance of blood flow by the tissue. So blood flow can increase 7 to 8 times as a result of vasodilation of the meta-arterioles and precapillary sphincters in response to increased rate of metabolism. Again, it makes sense. We are opening up those vessels and those sphincters in order to allow more blood to flow in into the areas that we are needing it due to the metabolism of that tissue. We have this term here, vasomotion. It is the periodic contraction and relaxation of precapillary sphincters. Blood flow through the capillaries. Blood flow will flow, my apologies, through capillaries until the byproduct of metabolisms are going to be removed or reduced, and the nutrients to the precapillary smooth muscles are going to be replenished. So we are going to have this vasomotion occur until we, I guess in a way, meet the demands of what that tissue is experiencing. The long-term local control is going to be capillaries becoming more dense in a region that regularly has increased metabolic rate. A great example I like to think of, because I had a lot of friends who ran track in high school, their legs always looked very, very vascular, in my opinion, and now we know why. And that is because you are going to have an increased amount of capillaries in regions where there is prolonged increased metabolic rate. So if you think about people who run or people who work out a lot, depending on those various parts of their body that they are exercising, they're going to need more blood flow in that area. If you have prolonged, I guess, metabolic needs needing to be met in those areas, think about training for like a runner or someone who lifts weights with their arms or whatever that may be. Those areas of their body need more blood flow. So they are going to create more capillaries in those areas to meet the demands of the tissue. So runners and people who work out will have more capillaries in those various parts of their body that they are working out more. All right, nervous control of blood flow in the tissues. These are going to be important in minute-to-minute regulations of local circulation. They are going to provide a means by which blood can be shunted from one large area of the peripheral circulatory system in another area by increasing resistance. The sympathetic division is most important and is going to innervate all vessels except capillaries, precapillary sphincters, and most arterioles. This is going to occur in and be controlled by the vasomotor center, which is going to be located in the lower pons and upper medulla oblongata. So think brainstem. I know that's probably way far back from previous semesters, but that is part of that brainstem. So the blood pressure must be adequate to cause blood flow through the capillaries while at rest, exercising, or in response to some type of circulatory shock. So if the BP, your blood pressure, decreases, we need to be able to respond to it. So when it comes to circulatory shock, this is going to be in response to blood loss, of course. So something is causing your blood to be exiting your body. There is not much blood there. So blood flow to your viscera and skin, so your organs and your skin, is going to decrease in order to help maintain the arterial blood pressure sufficient enough to allow adequate blood flow through the capillaries of the brain and heart. So this is kind of a cool response, in my opinion, that your body does when going into shock, essentially, is it will shunt, essentially, and kind of pick and choose what's more important to keep alive and keep that blood pressure up in that area. In this instance, it's going to be your brain. So as long as your brain's alive, you can survive, right? But you can probably survive without some of your extremities and maybe some of your organs. Another example is during exercise, the arterial blood pressure is going to be increased, and it is going to be needed to sustain an increased blood flow through your skeletal muscle capillaries, where precapillary sphincters are going to be dilated. We're going to have an increase in that blood flow and supply an increased amount of oxygen and nutrients to your skeletal muscles. So kind of two ways that this is going to be applicable here. Moving on, so the sympathetic stimulation of the adrenal medulla, and just to kind of give you a little heads up, the adrenal medulla is going to be part of your adrenal gland, which is going to be those glands that sit on top of your kidneys. There's a pair of them, one on each kidney. It is going to cause an output of norepinephrine and epinephrine into circulation. What I want you to think of when it comes to these hormones, which technically they're neurohormones, they can kind of be neurotransmitters and hormones at the same time, but that's neither here nor there. These are going to be the hormones that you release in response to stress or excitement, typically. But these are going to cause vasoconstriction in your vessels, except in skeletal muscle where vasodilation is going to occur. So everywhere else your vessels are going to constrict, but in your skeletal muscle, you're going to have more blood flow. And this is kind of a neat thing because it's going to be kind of, in a way, a fight or flight response. So we are increasing the amount of blood going to your muscles that are going to be able to get you out of there or fight you out of there. So kind of cool. And to finish up the slide, our constant sympathetic stimulation of vessels is going to result in a continual state of partial constriction, which we are going to call excuse me, vasomotor tone. This slide does show that nervous regulation of blood vessels and kind of how everything's kind of interacting with each other, but you won't need to regurgitate this slide in any way. All right, so we do have a few terms. There's only 4 that are going to have something to do with the short-term regulation of blood pressure. Some of these are minute to minute, and others are going to be a response to emergencies, and I'll tell you which ones are which. The first is our baroreceptive reflexes. So we've talked about baroreceptors before in this lecture, but they are changes in peripheral resistance, heart rate, and stroke volume in response to changes in blood pressure. So keep in mind that this is going to be stretch receptors giving back this information essentially. And this is going to be the most important of the reflexes that is a day-to-day type of thing. So this is one of our minute-to-minute responses. So these baroreceptors are going to be found along the walls of most large arteries that are going to be within the neck, and they are going to be sensitive to stretch. So specifically, just know that most of these are going to be found within the arteries within the neck. We have the adrenomedullary mechanism, which is going to be activated by a substantial increase in sympathetic stimulation of the heart and blood vessels, such as a sudden large decrease in blood pressure, substantial increase in exercise, or stressful conditions. You can also think of these as responding to some type of emergency, or specifically exercise. But this can also be in response to emergencies. The chemoreceptor reflex is going to be sensory receptors sensitive to oxygen, carbon dioxide, and pH levels of blood. These are going to respond to changes in your blood composition, if you want to think of it that way. So any of these things are going to change the composition of your blood in some capacity. Your chemoreceptors are going to pick up on that. So your blood oxygen can decrease in high altitudes, as well as just in environments where there's going to be higher carbon dioxide, lower oxygen. Your chemoreceptors are going to respond to that because it's obviously what you're taking into your body, right? From what's outside of it. The central nervous system is going to, the central nervous system ischemic response is going to result from high carbon dioxide or low pH levels in the medulla and increase the peripheral resistance. So this is specifically in your medulla oblongata. This is going to be a very rare emergency, and so the increase in blood pressure is going to be in response to a lack of blood flow to the oblongata, the medulla oblongata. But again, this is a rare instance, a rare emergency typically. I do want you to read through these various steps, if you will, for the baroreceptor reflex control of blood pressure. But in order to keep this lecture a little bit on the shorter side, I'm sure you can read through this. We kind of already talked about this a little bit. But yeah. All right. So the adrenal medullary mechanism. Again, this is something that you can kind of read through yourself. I don't want to bore you by just reading off the slide. I do want to make a little note here when it comes to the CNS ischemic response, and that is a reduced blood flow to the medulla oblongata. So decreased oxygen, increased CO2, decreased pH is going to cause the neurons of the vasomotor center to be strongly stimulated. It's also going to increase blood flow to the brain if the vessels are intact, but at the same time decrease oxygenation of blood because blood will not go to the lungs. So just kind of keep that in mind. And then the lack of oxygen will cause the vasomotor center to become inactive. Extensive vasodilation will follow with concomitant drop in blood pressure, and death will end up happening if the central nervous system ischemic response lasts longer than a few minutes. I think it's roughly 5 minutes that your brain can be deprived of oxygen. But your brain will die, essentially, if you're not able to give it the oxygen that it needs. So everything that we were talking about up till now were those short-term regulation of blood pressure. Let's talk about the long-term, or you can think of it this way, slow acting. So this is going to be a regulation of blood concentration and volume by the kidneys specifically. And then you have these various mechanisms, the renin angiotensin, aldosterone mechanism, and the vasopressin mechanism. And we'll talk about those here in just a second. But some mechanisms are going to respond in minutes, but continue for hours or days when it comes to these long-term regulations of blood pressure. And they are going to adjust blood pressure precisely and keep it within a narrow range of values for years, essentially. So it's whatever works for your body, it's going to try to maintain that to maintain homeostasis. Here we have the renin-angiotensin aldosterone mechanism, try saying that 10 times fast, and that process that it is going to go through in order to maintain blood pressure. Again, I'm not going to just sit here and read off the slide. You can read it just as easily as I can, and I'd like to keep this lecture under an hour and 30 minutes for you. All right, so then we have these long-term regulation of blood pressure terms that I do want to go over very quickly. This is the very last thing, but you have the atrial natriuretic, I'm going to try to say it, but it's not going to come out right. Hormone. It's a hormone, guys. And this is going to be released from cardiac muscle cells within the atria when atrial blood pressure increases, stimulating an increase in urinary production and causing a decrease in blood volume and blood pressure. The next is going to be fluid shift. Fluid shift is the movement of fluid from interstitial spaces into capillaries in response to a decrease in blood pressure to maintain blood volume and vice versa. And then last but not least, we have the stress relaxation response, and that is an adjustment of blood vessels, smooth muscle to respond to change in blood volume. And when blood volume suddenly declines and pressure drops, the smooth muscle will contract, and then vice versa. So I hope you've enjoyed this lecture. I'm sorry it is so long. I hope you have a wonderful rest of your day. Let me know if you have any questions.